Yeah,
Yes.
hello,
Hello.
and very warm welcome to the first episode of Expert Exchange 2.0, which is our exclusive webinar series designed to inspire, educate, and connect minds across the scientific community. Good afternoon to all joining us from India, and good morning to all our global audience tuning in from around the world. I'm Aanak Goswami from Acumex. I am honored today to be your moderator for today's session. At Acumex, we are proud to be more than just a product solution provider. Headquartered in Ahmedabad, India, with world-class manufacturing facilities across India and Germany, we have been at the forefront of laboratory innovation since 2003. Our portfolio today spans over 2,500 SKUs trusted in more than 130 plus countries, ranging from premium liquid handling systems to benchtop equipments and the bioprocess solutions. But beyond the products, our vision is to be your partners in progress, committed to sharing knowledge and empowering the next generation of researchers, innovators, and the problem solvers. That's what Expert Exchange 2.0 is all about, a platform when science meets dialogue and expertise is exchanged to drive the industry forward. Today, we are privileged to have with us two distinguished thought leaders from the industry, Dr. Varoon Singh and Dr. Sujata Sharma, who will be sharing their insights, experiences, and practical wisdom that can and inspire researchers at every level. Our first panelist today is a distinguished researcher whose work bridges the field of environmental science, biological chemistry, and precision medicine, Dr. Varoon Singh. Dr. Singh holds an MSc in organic chemistry from the University of Pune and he has earned his PhD from the prestigious DRD Gwalior, where he focused on advanced bioanalytical applications. Following his doctoral studies, Dr. Singh expanded his research globally, first as a postdoctoral fellow at the University of Waterloo in Canada and then at Ghent University in Belgium, where he contributed to cutting-edge research in precision medicine. He later joined the University of Vienna as a senior scientist, where his work centered on the development of analytical workflow and devices for single-cell metabolites, an emerging field with tremendous potential for personalized healthcare. Currently, Dr. Singh serves as a scientist at the National Drug Testing Laboratory in New Delhi. He plays a critical role in ensuring compliance with the World Anti-Doping Agency, WADA, by analyzing athlete samples for banned substances. Over the course of his career, Dr. Singh has authored 33 peer-reviewed publications, contributed 2 book chapters, and presented at numerous national and international scientific forums. He is also the named inventors on 4 patents, underscoring his commitment to innovation and cross-disciplinary impact. Sir, it's truly a pleasure to have you on our panel today.
Thank you very much for a very elaborated introduction. Thank you very much.
Our second panelist today brings an exceptional blend of academic leadership and industry expertise in the field of food science and nutrition, Dr. Sujata Sharma. With over 16 years of experience in teaching and research, including more than 13 years of experience as a professor of food and science and nutrition, Dr. Sharma has been a driving force in advancing nutritional sciences and applied food technologies. She earned her PhD from Agriculture University, Palampur, specializing in food science and nutrition. Dr. Sharma began her professional journey as a research fellow on an international project under the National Agriculture Innovation Project, funded by ICAR, where she contributed to Livelihood Society an initiative for tribal communities, a close cause to her heart. She then moved into the industrial space, bringing her academic expertise into practice. With over 7 years of experience in food industry, she served as head of research and development at FICCI Research and Analysis Center, where she played a key role in formulating and supervising policies related to food quality and safety. In recognition of her contribution to the field. Dr. Sharma was honored with a Lifetime Achievement Award in 2020 by the Center of Food Quality Assurance, New Delhi. She also holds a published international patent, further reflecting her commitment to innovation and applied sciences. Ma'am, we are delighted to have you with us today.
Am I audible?
Yes, ma'am, you are.
Okay, thank you so much. It's my privilege to attend this webinar, and I hope this is fruitful for us, for all of us, and maybe it's fruitful for the audience also. The audience will be interact with us, so it's really a great platform for us. I'm thankful to the Ecomax to inviting me here. Thank you.
Thank you, ma'am. So, for everyone on this call, we will go on the panel discussion with both our experts one on one with the first section of the panel discussion, which talks about why of calibration. So, ma'am, Dr. Sujata, I would like to start with you, with the basics. Why the pipette calibration matters so much in regulated and even non-regulated labs? What is your view on the pipe calibration?
Okay.
So, as we know that today the title for this webinar is Calibrate to Elevate, okay, for the ensuring accuracy. So it started with the pipetting. It's a volumetric methods. We generally use certain kind of tools to perform our technical analytical work. So calibration just we see with this is the piston operated volumetric apparatus, and it is also been considered with the IS methodology, where it is been very important to calibrate these kind of apparatuses. And we know that there are one standard which particularly based on this, and that is ISO 8655, and it has certain guidelines. That is they have again they have, I guess, amended it in 2024, and it is very important for all the laboratories, whether it is NABL accredited or non NABL accredited. Okay, so calibration itself gives you a significant results at the end of your analysis or at the end of your analytic work. And nowadays, if we talk about the scientific work, and it's really very, we cannot perform without this. So it plays a very important and crucial role in your task. And so this is, it has importance. On the
No,
other
ma'am.
hand, yeah, on the other hand, for the industrial or for the as I'm from the quality assurance field, and I do perform this kind of work for research as well as for quality assurance, means to quantify the to quantify something, or the quantify some kind of assess the type of assessment which we require. In that case also, this is a way where we verify our measuring, and it has certain standard operating procedures. We have to know about that, and we have to learn that. So volumetric, all the experiments need a calibration. That,
Absolutely,
that,
absolutely.
thank you.
Nicely voted, ma'am. Dr. Varun, what are your thoughts with the pipette calibration when it comes to regulated and non-regulated labs?
So, as I have worked in both kind of scenarios, I will try to tell my story from non-regulated first, then to the regulator. So when we are working in a research laboratory, we are trying to develop new methods. We are trying to showcase that, okay, our methods are capable of achieving limit of detections or limit of quantifications that are much better than the existing like FDA methods or methods laid down by the national regulatory bodies, whether these particular methods are able to achieve that particular LOD or not. So in that case, if your pipettes, wherever you are doing any kind of volumetric stuff, it is not calibrated, then maybe whatever you are trying to say that I have achieved a limit of detection of 0.001 nanogram per ml, maybe if it's not calibrated, it might be 0.01. And that plays a very important role if your pipettes are not calibrated, if you're not using calibrated instruments. So during my research days at university in India and abroad, I had privilege to work in ISO 17025 laboratories. So in our case, the pipettes needed to be calibrated once in a year in a research setup. But cut to the movement from non-regulated to the regulated laboratories, this becomes very stringent because now you are following an ISO standard for testing and calibration, which mandates you to follow ISO 8655 for any device which is using the pistons for calibration, and then it depends upon the number of samples you are analyzing and how frequently you are using these pipettes and how you're handling these pipettes. So in that case, some laboratories, they do calibration by third party three, 3 months or 6 months. And in some regulatory labs where the sample load is like 100 samples in a day or 200 samples in a day, it is mandatory for them according to their internal guidelines to calibrate their pipettes every day internally, and then quarterly by or yearly by a third party. So I think it's very important to make sure that to get an accurate and precise results, your pipettes are calibrated, and whatever results you are releasing, especially in terms of results that are held as a proof in court of law. So in those scenarios, your results highly depend upon what kind of instruments are you are using and whether they are calibrated frequently or not. So this is what I think is required to make sure that you are in a regulatory or whether you are not in regulatory laboratory, but the tools by which you are going to make sure that you are achieving these results are calibrated.
Certainly. And sir, you would also come across the cases where the calibrations are not done regularly, right? Or, when people rely on the pipettes for the long time without even calibrating it. So what would be the risk you feel, when the timelines of the calibration have been missed?
Okay, so in that scenario, what might happen that you have given a result. Say, let's talk a scenario where I have given a result for a particular case where uh, pers- a particular, for example, food. We have passed a food sample which is free from certain pesticides. We have tested them for pesticides, and our pipettes are not calibrated, and that particular food sample gets exported to another country. Their testing agency tests them, and then they say, okay, this food sample has higher residues of pesticides, but your laboratory has certified that it has lower levels of pesticides in it. In that case, what is going to happen? Your results are going to be challenged, and then you have to verify. And then when you will reach to your root cause, what will happen is like you will find whatever you are doing was case because of non-calibrated pipettes, and the person who was not trained to do internal calibration of your pipette, even if the person who is doing the internal calibration in your laboratory for maintaining the calibration, or I will say verifying the calibration, which was calibrated by a third party agency. And then in that case, scenarios will happen that your results are not validated, and then you will lose your credibility of testing, and you will lose the reputation as a lab. And that is going, if I'm a private laboratory, I'm going to lose my business. So this can have a serious impact on business results and the reputation of your lab and your country.
Yeah, true, true. Dr. Sujata, I would understand, like, obviously you would agree to these points what Dr. Varoon mentioned about the risk. Would you like to add something which you have come across with your experience, same purview?
Yeah, yeah, of course, of course. This is a very, very important for the laboratory as well as the analyst, food analyst, because it's a responsibility. It's just not the performing the task, because that sometimes we have a, product recalls, sometimes we have a certain you know mm, court issues. So that also related to the, with the precision and with the accuracy of the results. Not only with the negative part, but for the globalization of your product also required a certain, precise results for you. As sir said that residual, generally we analyze residual effect at the PPP level or the micro level. Again, if accuracy is not there, your the apparatus, the chosen apparatus and the performance not ensuring the accuracy in your task. Again, it is a good big challenge for the that laboratory. Secondly, I would like to add in that, like, we generally have audits. So we have internal audits and we have external audits. So calibration itself is a big non-performing activity if you are not calibrating it. Okay, it raise NC generally, and it is ignored by the laboratory persons. They generally forget it to calibrate, or they do it by their own self. But there is no validation. So validation is also very important with the third party, and calibration with the standards, the laid down standards, and with their requirements that needs to be met. So audits, during the audit, is very helpful. Yeah.
True. What are your recommendation? If we talk about, we've talked a lot about the why pipette calibration is important, right? So what would be the best practices you would recommend, for the high use labs versus the occasional setups for the pipette calibration?
So it's based on the organizational standard operating procedures, and it is based and it is vary with the laboratory to laboratory, or with the we have a microbiological performance laboratories. We have a means it's vary. It's depends on the uses also. So some calibrated apparatus or instruments are generally calibrated uh, by their persons, and some follow the guidelines given by the standards, and that is also included with the ISO 17025, and it's a part of the laboratory task. Yeah.
Right. True, man. Dr. Varun, your views on the calibration frequency recommendation for the high throughput lab? How often should they, get their pipettes calibrated?
So in terms of like laboratories that are ISO 17025 and have higher frequency or like high throughput samples, uh, in our laboratory, I will say we do calibration every day, internal calibration, and that is traced back to calibrated weights that is bought by us or from laboratories that have traceability to international standards in SIN. So we use calibrated weights for calibration of our pipettes, and we do it by gravimetric method. We use water and we measure water and the weight again, and then we compare we have a validation sheet for pipette calibration. We measure certain replicates, we see, and if the results are within the calibration range, which is provided by the third party, and we compare the deviation. If the deviation is within the range, then it is good. And if it is not within the range, we are not using the pipette until it is calibrated properly. If there is problem with pipette, we generally go for like cleaning. There is a piston cleaning of check whether the O-rings are fine or not. And if that also doesn't clear the problem, we send it for recalibration. And it depends. If you are doing calibration every day, internal calibration every day, any deviation will be marked in your results. And the moment it is marked in our results in pipette calibration, we isolate that pipette and then we send it for calibration. Because we are a laboratory where the results are held as a proof in the court of law. We are doing athlete samples testing, and a result provided by us can become a life decision for an athlete because his career or her career or their career depends upon the results which we are providing. It might bring ban on them, for a long time. So, and we have to defend our results. If we can't defend our results in court of law, then in that case, we are not willing or we are not comfortable releasing a report which can put us in trouble. So, pipette calibration in our case is a very serious matter. Our analysts are trained to calibrate the pipettes every day in the morning. And there is one senior person who verifies that the calibration has taken place in front of them. It's not like that a trainee or an analyst has put down the results from previous day and then somebody is signing. No, there is one person who is overseeing this calibration and then it is getting signed. So as to make sure that a small thing which can have a larger impact on the results, on the laboratory, on the nation, does not create any havoc. So I think having a routine calibration every day in a labs where 200 samples or 100 samples are handled manually. So having an internal calibration every day is very important, and I suggest doing that.
So I think, sir, your lab does take utmost care for the calibration, and it is not just an alternative, but it is a compulsion. So which is a standard which you guys follow. What would you suggest, sir, for day-to-day care? Like, what would be that one thing which every lab user should do daily to extend their pipettes accuracy and the maintenance?
Handling of pipette.
Yeah,
That
yeah.
is that is the most important thing which people forget, because when you are doing a lot of things in a single day, you are trying to achieve your number of samples in a day. What you end up doing is like to increase your throughput, you compromise with the accuracy and the way you are handling your pipettes and the samples. What happens is like people, I have seen my analysts doing this mistake. So whenever we have a new analyst, we test them first on how they do pipetting, because that is the core of our testing. So what we generally suggest them during our training, that you have to keep your pipettes straight every time when you are doing pipetting, dispensing your samples. Even if you have a solvent or a liquid inside your pipette, do not tilt your pipette more than 5 degrees. If it has to be 90 degrees, make sure it is 95, 85, but don't go beyond that. The liquid can go inside, can ruin the pipette's O-ring, can ruin it can do the aerosol contamination. It can have things getting stuck inside the pipette chamber. So it can create problem. I have seen this is the major mistake done by analysts while pipetting and handling samples. They tilt their pipette with solvent, and then that creates whole problem during the testing and analysis procedure. The second is not keeping the pipettes on the pipette stand,
Right.
because once they are used, they keep it on the stand just by keeping it horizontally instead of keeping it vertically at their right place. Because if they are not kept on the right position with the right, I will say, the markings on it, sometimes people prefer that the pipette should be put on full volume side, like 1,000 microliter. If it is 1,000 microliter pipette, the spring should be released to 1,000 microliter and then it should be kept on the pipette stand. Some people prefer it should be always under strain. But if you know the spring, the Hooke's law and everything, the spring and which is going to push your sample out or keep it inside, has a very important role on the accuracy of your sample. So if you keep it always in the, say if it's 1,000 microliter pipette and you keep it at 500 microliter every time, the spring is always pressed. It's not coming to its original shape. It might affect your accuracy. And if it is always at its full volume, then it has its original shape. That's what I generally tell my students and my analysts, that make sure that your pipettes at the end of the day are set to the full volume range and they are stored in like that position.
Absolutely. Dr. Sujata, what are your views? And to add up on what Dr. Varun mentioned about day-to-day care, what would be those things which wherein a researcher can improve the pipette life as well as the accuracy of the pipetting.
Yeah, as Varun sir said that it's really really very important because small deviations have significant consequences. Am I audible to all of you? I feel there is network issue. No, it's okay.
Audible,
Thank you.
ma'am.
Okay. So, in a laboratory research, handling of equipment or handling of every small tool is very important. And sometimes we have to train them because I work with the students, so students are not the analyst. They don't know the procedure of testing, they don't know about the ISO or something. At laboratory person it's true, they know that everything it's very important, but at research institutes or in as a academician, we have to teach them that you have to use this uh, pipette or this calibration like this. You have to do the procedure, you have to follow these all steps before performing any test in on a instrument. Or generally we have methodologies laid down by the researchers, so they follow that methodology. Sometimes we deviate that methodologies also because results are uncertain, and they just want to research on the same aspect. So in that case also, the set pattern they need to follow, because deviation always gives you a wrong result sometime, or maybe the desired result you will not able to get that, and the same you will interpret on the data, and data will be wrong. So that will impact on research also. So we generally take care of this all. We have technical persons, they are trained, so they are skilled for these all laboratory practices. So they teach them, and under their supervision they work further on instruments. So yeah, thank you.
Are there any red flags according to you user should notice themselves before the performance issues arise? Because a lot of time
Sure.
we are not able to notice the issues. So what are those red flags you suggest?
So for example, if you talk about the weighing, so weighing means how accurately you have taken a sample, and what are the measures around that you have taken care. If their fan is on, again you will not get a accurate weight of that sample. Even there are many issues we have seen as a red flags when we see the results come out from the repetition, or the with the consistency of the same procedure. Again we see there are deviations, there are no significant values we found in that, so it means the results are not accurate. So again, the causes, root causes may be the handling of that instrument, or maybe some technical issue with that. So it's very very important, and it's really really very very significant to interpret something that is based on your analytical work. Yeah,
True, true.
because we generally in research, no, we compare our results with the others. So if someone has quoted it, for example, if someone has quoted like this sample is giving us 19% protein, okay, but when you are analyzing your sample and you're not getting it is within the range, it means there are something lack. Means you are not, maybe the sample is not correct, or maybe your handling of that sample is not correct, or maybe the analytical part of that sample you have analyzed is not correct. So this is significant, and this is really really very important. So we cannot just perform a task. We have to see that all the, it means to get an accurate task or the desired results, you need to be very skilled to handle this kind of apparatus. Yeah, thank you.
Dr. What I mean, I would like to have your suggestions on internal versus external calibration. When is in-house calibration sufficient, or maybe when should labs rely on certified third-party calibration? As you mentioned, like with your lab, your scenario, you do it in-house day to day, and obviously periodically you would be sending it out. So what should be the balance of the things? Obviously it will matter in different scenario differently, but what is your take on this?
Like if you are doing, if you're not a regulatory lab, calibrating external calibration once in a year is more than enough. But then if you're not able to do internal calibration every day, then maybe bi-weekly is good if number of samples is okayish. But if I say you are handling a lot of samples, frequent calibrations, if not every day, then once in a week should be recommended. It is also part of standard operating procedure of that laboratory, that what is their SOP stating that how do they have to calibrate and which method they have to use for calibration, like gravimetric method, photometric method, whichever they prefer. Most laboratories I know, wherever I have worked, they prefer gravimetric method because water is same everywhere and every lab has a microbalance. So they can have similar results and easy to train people on doing internal calibration. So depending upon the number of samples and type of laboratory and the place where these pipettes are being used, it can be decided by the laboratory manager and the professor who is handling the lab, uh, that what will be the frequency of internal calibration. But suggesting from my point of view, having external calibration once in a year is highly recommended. And also, while doing internal calibration, people should take care of certain things: humidity, temperature, because the weight of the sample, say water, is going to depend upon the temperature also. As whenever we are doing calibration, we have to make sure what is the temperature, and it should be written at the time of calibration, because in India that varies a lot. And because we have regions from north to south, temperature variation is very high. From east to west, there is humidity problems. So humidity below 50% creates problem, because then evaporation will be fast. Humidity at higher level is okay, but if the temperature is 50 degrees in Rajasthan and around 5 degrees in Jammu and Kashmir, it is also going to affect your internal calibration. So you have to make sure that these things are taken care of while doing the calibrations.
Very true, sir. Dr. Sujata, what documents, or certificates you think that customers should ask when they are, getting the calibration done outside their lab? So what are those things which customers should keep in mind when they are getting their pipettes calibrated on the third party setup?
Yes. No, it's not from outside the lab. They came to the lab and then they calibrated with their own
Okay.
procedures. Yeah.
Okay. So what are the documents?
Generally,
Uh,
just wait. Yeah. Generally, it is already calibrated. The operators are already have measures. Already it is calibrated. But while you are using it again and again and again, and then you require a calibration, okay, or the cleaning also, not of the calibration but the cleaning is very very important in that. So this is handling of your laboratory practices, means how you handle your analytic work at laboratory. This is very important to learn to the student or to the researcher or to the analyst who is working for that. Your question was how it is.
So
How?
I need to understand more on, once you got the pipette, obviously the documents comes along with it and it is
Yeah.
calculated. And
Yes.
after 1 year or after 6 months time, when you're looking for a calibration to be done outside the lab, or you're sending the pipette somewhere for the calibration. So what are those documentation, or, the customer should keep into the
Yeah.
mind when they should send the pipette?
Actually, every organization is under some standardization or ISO they are following, right? Some are following in a food sector, because I am expert with the food sector, so I will talk about that and I will quote example with the food sector. For example, ISO 22000 is there, right? So ISO 22000 has certain clauses. and with that clauses you require to document and to record everything. So record is based on the practices, whatever kind of practices you have been there, means that may be the small, like you have a logbook for the temperature, you maybe have a logbook for the weight, you maybe have a documentation for procurement or everything. So this is a part of that document. Okay, so calibration is also plays a very very important role in that. Like if you are weighing something or you are using something, how many times you have used that, and how you have, what are your practice to use that. So it's a part of your standard. Secondly, it's a part of your quality manuals, it's a part of your auditing part, or means it's a really really very very important aspect in that. And third party audit is mandatory in there, this kind of standardizations or this kind of organization, because it's a again, auditing means for the validation. So they cannot validate their procedures or the practices by themselves, so it's required to be validated by the third party who have that standardization. Yeah, yeah.
Very, very true, ma'am. And I think we are having good insights for both the expert panelists on the room today with us. So with this, I think we have ended our panel discussion part. We would like to take question and answers, questions from the attendees, if any, and we can take the questions one on one. The session is open for the Q&A for the people or for the participants who are here in the room. You can also write it on the chat box, or else you can unmute yourself and have questions if you have. Okay. Yes. So I think we have one question. Allow me to read. So how important is the ISO 86556 2024 standard? Dr. Varun, you would like to take this?
Yeah, sure. See, it's very important. It's a part of the 17025. If you're a 17025 accredited laboratory or any ISO following laboratory, I'm talking for all the laboratories that are involved in testing and calibration. They need 17025. And if you are following 17025, you need your pipettes to be calibrated by a third party. And once a third party is calibrating, they need to follow ISO 8655 in order to calibrate your pipettes. So if you are getting your pipettes calibrated by a third party, you are actually following 8655. And then on top of that, you are doing internal calibration. That is also following ISO 8655, because while doing calibration, you have to take care of all the clauses laid down in 8655 for pipette calibration. So it's very important for testing and calibration laboratories, be it for food testing, bioanalytical, clinical, any kind of testing laboratory, they need 17025. And the moment they go for 17025, they are immediately following 8655.
So, ma'am, there's one more question which I can read here, which talks about what are three best features or three best points to consider in the mind as a user when you are purchasing a pipette. Dr. Sujata, would you like to take this?
So users always see the convenience first, means whatever kind of apparatus they want to use, it should be convenient to handle. Okay, and their cleaning is also important, and the accuracy is very important. So being a consumer, when I have to choose something, I will choose these kind of material according to my need. Okay, but generally it is based on the measurements. So it's based on the measurements. What are your requirement? You will use that. Okay, that could be micro, that could be in grams, that could be according to you. But generally, this piston operated are with the minute measurements where you require a micro level measurements. It's very easy to handle because you don't need anything. So pipette is playing a very crucial role as well as significant role. So we availability is also very important, that we in India, if I need something, it should be available within India, because cost is also important part while we are purchasing it. Yeah.
Before we wrap up, there's one last question which I can take is what are the key differences between calibration and verification? Is this person asking what are the key differences between calibration and verification? Dr. Varun, you would like to take this?
Definitely, I will, because I use this word during explaining things. So calibration is like you are verifying you are actually calibrating the instrument according to 8655, and you are measuring its accuracy at lower volume, which it can take, higher volume, which it can measure, and then you are also getting the uncertainty calculated for this particular measurement. These are the three things which you should also look in your external documents. That what is the lowest volume you can measure, what is the highest volume you can measure, what are the uncertainties associated with this measurement, and this is the part of external calibration, and this includes everything in calibration. But during verification, what you are doing, whatever results you have got from your calibration partner, are you able to get the same results? Are you able to get the same uncertainties while doing the verification of your pipette calibration? So it is kind of verification of already acquired data using your instrument, but you are not calibrating the instrument. You are just verifying the values. But in terms of calibration, if there is some offset value, say I had to measure 1000 microliters and my pipette is getting 990 microliters. So in that case, what should be done to calibrate this volume, or what other measures can be taken to take to offset this value? I do not want to calibrate. I have verified my results are not accurate, but I can offset this value. So if I know that my pipette is calibrated, taking only 990 microliter, then you can then offset for this 10 microliter less volume. And then also you can get the accuracy. But these things are done when it is impossible to calibrate, or you are not able to calibrate on time, or your results are very important and you have to release it within hours, and then you can do this, and this will need a deviation from your SOP and approval from quality manager and quality assurance department. So these are the things which are considered under verification, and calibration is completely different.
Very, very well explained, Dr. Varun. And before I wrap up, one last request, which is already there. How important, it is to have an ergonomic pipette, and how can we avoid RSI by having an ergonomic pipette? Dr. Sujata, would you like to have this on the ergonomics of the pipette? Since you've already spoken on those points, would you want to take this?
I feel Varun sir will answer it well, because he has a good experience in that, and he's detailing the all the matter. Yeah.
I will take the question. So the ergonomics do play a role, because when I have to pipette 200 times in a day, and if it is creating a problem during handling, so it should be easy to handle. Ergonomics, I will say, some people have small fingers, some people have large fingers. That also plays an important role, that when you choose a pipette design, you choose it according to the people who are working in your laboratory. But right now, I think it's always standardized, and the pipette should be easy to hold, and your fingers should be easily reachable to the top of the. Uh, I'm talking about microchannel pipettes. So there are different pipettes, you have positive pressure, you have negative pressure, depending upon use. So if you are using it twice in a day, it doesn't matter. But if you are using it for 200 times in a day, then ergonomics do matter. It's like sitting in a chair. If it's very comfortable, you can sit in a chair for 4 hours. But if it's not comfortable, you will like to get up after 10 minutes. So, yeah, it does matter, but you have to decide because your budget also tells you what you can have. Microchannel pipettes are not cheaper, they are expensive. Some labs will prefer to have cheap but rugged instruments or pipettes. So, yeah, do. If you are rich enough to have a ergonomically stable and nice pipette, go for them.
I would like to add some few points in that which has been ignored, I guess, by the manufacturers, and that is the weight of pipette is very important. Secondly, the material they are using in that, because we have to discard this material, and they generally use a rigid material in that, and the discard of pipette and the laboratory materials which is being discarded, it creating a lots of waste. So this is also very important part to be think of, that what kind of laboratory materials are there and how to discard them, because after some time we discard these kind of materials, and there are non-recyclable, or no one is there to collect that. They've been treated as a plastics. So it's very important.
Absolutely. And just to add, like what you mentioned and what Dr. Varun also mentioned, ergonomics plays a very crucial role for pipetting accuracy as well as the well-being of the person who is pipetting repeatedly. If it is only 2, 3 times a day, it's fine. But when it is pipetting multiple samples, it becomes very crucial for them for their own thumb well-being. And it's very important to have a pipette that has a low plunging force as well as good ergonomics material, good ejection force rather to to choose pipette according to those requirements, obviously. So Dr. Varun and Dr. Sujata, I mean, it was pleasure to have you guys here, and it was deep insights which we've got from you guys on this topic. I'm thanking you at the level of it was a pleasure having you around, on this discussion, and I hope this session would have given few key takeaways to the viewers who have joined us in this session. So thank you so much, sir. Thank you so much, ma'am, uh, to be with us on this session. And for the people who are there on this call, I would just request you to stay tuned for more such sessions coming your way in this Expert Exchange 2.0 webinar series. Thank you for joining. Thanks a lot.
Thank you very much for this opportunity.
Thank
Thank
you.
you.
Yeah.
Thank you.
Thank you very much. Thank you.
Red color